I'm from Cincinnati Children's Hospital Medical Center. I will talk about the project to generate pulmonary endothelial progenitor cells from cell-based therapy using interspecies mouse rat chimeras. So the lung morphogenesis is dependent on the formation of capillary network that requires a sustainable differentiation and proliferation of endothelial progenitor cells. c-KIT had been found to be a representative marker for endothelial progenitor cells. The published result by our lab found lung capillary network compromised in FOXF1 heterozygote mice. Recently, we identified that c-KIT positive endothelial progenitor cells specifically reduced in FOXF1 mutant mice. Here, you can find from the flow cytometry analysis that in wild type and in FOXF1 knockout mice, the c-KIT positive endothelial cells significantly reduced. So in both hyperoxia condition and room air condition. Also, the deficiency of c-KIT positive endothelial progenitor cells cause alveolar simplification in FOXF1 knockout mice. The c-KIT positive endothelial progenitor cells are conserved and widely distributed in capillary bed in both human and mouse. Here you can find the co-staining of c-KIT with the endothelial marker PKM1. And to understand the heterogeneity of pulmonary endothelial cells, we run single-cell RNA-seq analysis on endothelial cells isolated from P7. It was found that c-KIT positive endothelial cells highly enriched in a subcluster. To understand the functional relation of c-KIT positive endothelial progenitor cells with alveolar formation in FOXF1 mutant, we developed a transgenic reporter mouse model with CRISPR technology. In this mouse model, GLP was inserted into the endogenous FOXF1 locus, and the co-staining GLP with the FOXF1 antibody indicated that this reporter mice recapitulate the expression pattern of FOXF1 very well. Another mouse model, SVV2 mutant, contains a single point mutation in the transcription activation domain of FOXF1. It also developed characteristic phenotype in FOXF1 conditional knockout mouse model, like the hemorrhage, fusion of lung lobes, and alveolar simplification. Next, we isolated the FOXF1 positive and c-KIT positive endothelial progenitor cells with flow cytometry sorting for rescue assay in SVV2 mouse model. Here, confocal analysis demonstrated that the cells are effective in improving the phenotype of alveolar simplification. Next, we derived the FOXF1 positive, c-KIT positive endothelial progenitor cells for cell-based therapy with the interspecies mouse rat chimera. First, we generate an embryonic stem cell line, which express both FOXF1 GLP and the TD tomato. And this stem cell line was used to generate the FOXF1 positive and c-KIT positive endothelial progenitor cells, while a blastocyst complementation. The flow cytometry was used to isolate the stem cell derived progenitor cells. And the red panel had shown it was found that the endothelial progenitor cell derived from stem cell is equally effective as the endogenous progenitor cell, can also rescue the alveolar simplification phenotype. So here is the brief summary. So to further characterize the FOXF1 positive and c-KIT positive endothelial progenitor cells, we run parallel single-cell RNA-seq analysis of pulmonary endothelial cells derived from interspecies mouse rat chimera together with the wild-type control endothelial cells. The left panel is the integrated projection of endothelial cells from chimera and control. As shown in the heat map, it was found that the gene expression pattern of endothelial cells derived from the chimera are very similar to the control endothelial cells. So here's a brief summary. I identify and characterize of the FOXF1 positive, c-KIT positive pulmonary endothelial progenitor cells by integration of CRISPR technology, flow cytometry, and single-cell RNA-seq analysis. So next, we generate the FOXF1 positive and c-KIT positive progenitor cells with the interspecies mouse-related chimera by blastocyst complementation. So thanks to the guidance and instructions from Dr. Vladimir Kalinchenko at the Department of Pulmonary Biology, and also affiliated with the Center for Lung Regenerative Medicine, or from Cincinnati Children's Hospital Medical Center. Now I'll stop to take some questions.